Good afternoon. This is Friday, October 2nd, and I'm going to introduce you to the pH and Buffer Lab for AP Biology. So you can go into Google Classroom and open the Google Doc, or you can print out a copy. Oh, that's not working too well. Print out a copy. I'm going to have to take this background off. It's a little sunny behind me. That's why I put the background in, but let's take it out. So anyway, get a paper copy, print it off, or use the online Google form. Either one's fine. Take a moment, once you have this, take a moment to read the introductory information. Read the part 1 refresher on pH. And then if you would take a moment and make predictions, center column, make predictions about these 4 substances and their pH. So we had a brief discussion about pH and how pH is determined by the dissociation of water. On the learning objectives, there's a video by Bozeman on acid, base, and buffers. I suggest you watch if you are not that familiar with pH. But during most reactions, a small amount of water dissociates, which means it breaks into ions of hydrogen ions, H+, and OH ions, OH negative. By the time the reaction concludes, oftentimes you either have extra hydrogen left over or extra hydroxides left over. If you have extra hydrogen left over, that contributes to making the solution, the surroundings, more acidic. The pH drops. Whereas if you tend to have more hydroxide OH negatives remaining in the environment, that does the opposite. That makes it more of an alkaline or base, and the pH rises. pH is absolutely critical to many biological functions. It's one of those things that we must maintain balance, homeostasis for. And so in this lab, we're going to investigate how do living organisms maintain a balanced pH? How do you maintain a stable pH? Because later on, we're going to find out pH has a huge effect on protein folding and protein shape, which means it affects protein function. So if your pH is not remaining stable, your proteins aren't going to remain stable. They're not going to function properly, and you're going to be in a world of hurt. So anyway, stop the video, take a moment and read that introduction, and go ahead and please make your predictions, and then we will come back in a moment and take a look at the results. Go ahead, pause. Welcome back. So I'm going to move my screen down a little bit. So I have what's called a well plate. And in this well plate, I have 4 solutions. I have the milk, which is obvious in white. I have apple vinegar cider, apple vinegar. I have bleach or ammonia, and I have plain water. So I'm going to take some pH paper. You've probably worked with pH paper before. pH paper, you put it in the solution, it turns color, and then you match the color to the scale. And that gives you the approximate pH value of what you're looking at. So milk, if I go in with my pH paper, so I've just got a little strip of pH paper. I'm going to put it into the milk. Let it sit for a moment, bring it out, give it a moment to change color. And you can see that that matches up to pH 7 pretty darn well. So milk actually has a neutral pH, pH 7. Grab another sheet, wipe off my tips of my forceps. I'm going into the vinegar. Let it come out. And I can see I'm getting a very different color out of the vinegar. That's going to be much lower on my scale. And if I go to match it up, I'm getting right around pH 3. Somewhere between 3 and 4. I'm going to call it a 3. So pH 3 for vinegar, which makes sense because vinegar is known to be very acidic. Test our bleach or ammonia, which many of us know can be quite dangerous. And we see we get the other end of the spectrum. Bleach has a pH somewhere around 9 or 10. Looks like it's a little bit more of a 9 in this case. Some bleach solutions are much stronger. Some will go all the way up to 12s. But this one's a 9. And then finally, we just have good old plain tap water. And I'll stick this into the tap water. Bring it out, let it set for a second. I'm going to find that matches up pretty well with pH 6. Okay, so that's just the pH of some common materials. So now we're going to go into the experimental part of this. So I don't need the well plate anymore. What we did, we're in teams of 4. So 2 people took on the acid test, 2 people took on the base test. It's really the same test. It's just you're adding a different solution. So in each case, we started with water. So if you're looking at page 2, there's data tables, and water is the first one listed. There's the first data table deals with adding the HCl, which is the acid, the hydrochloric acid. And the second table deals with adding the base, the NaOH or sodium hydroxide. So I've pre-measured 10 mils in my graduated cylinder. There's 10 mils of water. And I've got a second one that I also measured 10 mils of water. And we'll put that in the beaker. So let's do the acid one first. This is a weak acid, 0.1. It's HCl, hydrochloric acid. It ionizes into hydrogen and chlorine, so it adds hydrogen to the solution, thus making it more acidic. So we're going to take the initial pH at 0 drops. I'm just trying to pull these pieces apart. And at 0 drops, we've got a pH of, it's right in between there, but this looks more like a pH of 7. So 7 with 0 drops. Then we're going to add 10 drops. These drops will come out drop by drop. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We're going to swirl that around and mix it. And then we are going to take a reading. And I can see that it's turned a little bit reddish. And boy, we are already down to a pH of 2. So 10 drops puts us down at a pH of 2. And I'm going to add 10 more drops. 9, 10. I'm going to swirl that again. I'm going to take the pH. And that's become even a darker red. That's close. I'm going to call it pH 1. And 10 more drops, can't take it any lower, but 2, 3, 4, 5, 6, 7, 8, 9, 10. And I will take my forceps and drop this in. And this is a very deep red. And there's definitely still a pH of 1. So we went from 7 to 2 to 1 to 1. Now we do the same thing with the other beaker, adding our base, NaOH, sodium hydroxide. That's going to contribute sodium ions and hydroxide ions. Extra hydroxide ions will raise the pH and make it more basic. So again, I'm going to do an initial pH. I may use a different set of forceps. And again, I'm getting more of a 7. I'm going to add the first 10 drops of the base. 2, 3, 4, 5, 6, 7, 8, 9, 10. Put that in, and I'm getting pretty blue all of a sudden, really quickly. And I'll call that a pH of 10. Add 10 more drops. And I got to get some more pH paper out. Swirl that around. And so now this is 20 drops. And you can clearly see it's coming out quite blue. I would say that's at 11. And finally, 10 more drops. Total of 30 drops now. Swirl it around. Take it out, even a darker blue, and that is up into the 12 range. So 7 to 10 to 11 to 12. And so water is representing a non-living system. Water has a non-living system, and this is showing water has no natural capabilities to combat changes in pH. If you keep adding acids, it's going to become more acidic very quickly. If you keep adding a base, it's going to become basic very quickly. Has no resistance to change in pH. Living things can't do that. So what do living things do to combat changes in pH? So to do that, and we didn't get this far in class today, we are going to repeat the exact same process, only instead of water, they're going to do a potato homogenate, which is basically potato ground up in water. So when you grind the potato up in the water, you're breaking open the potato cells and all the cell contents are getting into the water. So it's representing the chemicals that you would find in living potato cells. Then we'll dump it and we'll repeat again with a liver homogenate. Using chicken livers, we'll grind it up. That again will dump the cell contents into the liquid, and we will see that is our our animal example. So potatoes are our example of how can plants respond any different than plain water? Chicken livers are our example. Do animals respond any different than plain water? And then we're going to test this thing called a buffer. And our buffers, what do they do? Is there any connection? So we're going to do 3 more tests: potato homogenate, liver homogenate, and buffer. And then from that, you should be able to start making some claims and drawing some conclusions. So record the water data if you haven't already. And we will share the data from the class that we get on Tuesday for the potato, the liver, and the buffer. And I believe I've reset this not to be due until Tuesday night. Okay. And there's some graphing you'll need to do, and there's a CER that you will need to do. And you'll get more instruction on that as we get closer to Tuesday. All right, it's Friday. Enjoy that weekend.